An apple tree, a daisy, a carrot, a cornstalk, or even just a piece of clover. All of them start from something startlingly miniature. A seed is basically a dormant vessel, a suspended organism awaiting a catalyst. But how on earth does a single rigid husk contain the biological architecture for a massive, centuries-old tree? There is a fascinating story about a Russian research team that recovered seeds of Silene stenophila, a flowering plant native to Siberia, from deep permafrost deposits. They were found in the stash of an Ice Age squirrel. An Ice Age squirrel, imagine that! And through radiocarbon dating, scientists verified that those seeds were 32,000 years old. Just think about that temporal scale. 32,000 years of glacial silence, and yet the complex biological machinery trapped inside remained viable. Today, we're going to dissect the intricate systems that transform a minuscule speck into a towering forest. Now, beneath the surface of the Earth, the environment is far from empty. It's more like a subterranean waiting room. A seed rests in the substrate, utterly still. It has no leaves to capture solar energy, and it's got no root system to pull minerals from the soil. Yet the organism is not dead. It's in a state of suspended animation. For germination to actually happen, the seed needs a precise alignment of variables: moisture, the right temperature, and a specific mineral composition in the soil. If any of these conditions are missing, the organism remains locked within its fibrous exterior, a prisoner of its own environment. This process isn't just some random chance. Plants have evolved adaptations to specific habitats. For example, a desert acacia would simply perish in a temperate rainforest, and a high-altitude conifer cannot thrive in a grassland. The seed actually retains the precise geographic parameters for its successful development. It's essentially a biological machine programmed for a specific coordinate on the planet. If you want to see this transformation in real time, just look at the mechanics of the common pole bean or lima bean. These varieties have incredibly rapid development cycles. If you place them in containers, whether it's salvaged eggshells, tin cans, or even just terracotta, they can complete the transition from a dry husk to a multi-leafed organism in just a few weeks. The only thing is, drainage is vital. Without holes at the base of the container, the soil becomes waterlogged and you get rot instead of growth. If you organize a dozen eggshells in a carton, put them on a windowsill where the sunlight is consistent, and number the containers so you can monitor them closely, the progression becomes measurable. You put one bean in the center of each, give them a controlled sprinkle of moisture, and you initiate the sequence. It's a tactile, methodical procedure. And witnessing the rapid unfurling of the bean shoot is like watching a biological acceleration in fast forward. External pressure is what starts the shift. The dormant vessel, which was previously static, begins to draw moisture from the surrounding earth. This is the primary stage of plant development, imbibition. As water saturates the internal tissues, the volume increases, and it starts exerting an outward force against the protective husk. This tension, this pressure, causes the exterior layer to soften and eventually fissure. This rupture signals the start of germination, the transition from a dormant entity into an active, growing organism. But this isn't a random occurrence. It's highly regulated. It's governed by a precise interplay of physiological, molecular, and environmental factors. The seed doesn't just wake up because it's wet. It requires a specific chemical signal to launch the process. It's a sophisticated biological machine engaging its systems for the very first time. If you peel back the outer layer, you'll find the standard components of this survival kit. There are three primary elements: the protective boundary, the embryo, and the cotyledons. The outer layer is basically a fortified shield, keeping the internal structure secure against external contaminants. Inside that shield resides the embryo, the nascent organism, a microscopic, perfectly formed blueprint of the eventual structure. And adjacent to this are the cotyledons, which house the internal nutrient stores, often containing the endosperm. This reserve is absolutely critical because the embryo lacks the capacity for photosynthesis until it actually breaches the surface. Chemistry then dictates the next phase. The internal nutrients activate enzymatic reactions that provide fuel. These enzymes catalyze the conversion of stored energy into forms usable by the growing embryo, allowing it to penetrate the substrate. The entire structure is optimized for one single purpose: achieving the surface to begin autonomous feeding. Now, here is where things get a bit strange. Certain seeds simply refuse to germinate, regardless of how much moisture or light they have. This represents a bizarre paradox in botanical science. Why would a plant maintain a state of dormancy under seemingly ideal conditions? The answer lies in environmental cues. For instance, seeds of the Protea species from South Africa only germinate after exposure to smoke. Smoke! This is a calculated response. The Protea requires the aftermath of a wildfire to thrive. By waiting for the chemical signature of smoke, the seed ensures that its competition, the surrounding vegetation, has been incinerated, and the soil is now enriched with ash and devoid of shade. It's not an arbitrary delay, but a high-stakes bet on the most volatile event in its environment. Inside the microscopic world of the seed, a molecular tug-of-war is happening. While water and smoke provide the environmental triggers, the internal mechanics are driven by two opposing forces: abscisic acid, or ABA, and gibberellins, known as GA. This interaction is like a high-stakes biological standoff. When the chemical balance shifts, the metabolic acceleration begins. During the final stage of germination, seed cells activate and elongate. The protective layer ruptures further, and the radicle, the primary root, emerges. This is the point of no return. The seed is no longer a waiting room, it's an active seedling. Internal pressure continues to rise until the outer husk splits entirely, preparing the organism to face the environment above the soil. Plants utilize varying survival strategies to maximize their generational success. Oak trees, for example, exhibit a much slower, more deliberate growth trajectory. This is a risk management calculation. If an oak seed germinated at the first hint of warmth during a mild winter, a subsequent frost would destroy the shoot instantly. So, by staggering germination over decades, these trees ensure the continuity of their lineage. The scale of this patience is just immense. An oak tree may continue its cycle of growth while generations of humans pass through their entire lifespans. While bean plants prioritize speed, the oak plays a long game, diversifying its timing to withstand unpredictable climate shifts. This bet hedging prevents the entire population from being wiped out by a single adverse season. Seeds often appear as inert pebbles, but they function more like time capsules. The realization that they are actively monitoring their environment, waiting for the precise microsignal to initiate growth, is profound. Observe how the nascent plant secures its future. The radicle drives downward, deep into the substrate, seeking moisture and mineral stability. This is not merely growth, it's the establishment of an anchor. The root system locks the organism into the earth, preventing displacement by external forces. It's a precise, autonomous machine working in total darkness, converting a microscopic packet of energy into a permanent fixture of the terrain. Once the root anchors the plant, the ascent commences. The young structure breaches the protective layer and rises toward the surface. It pushes through the soil as a pale, fragile shoot, driven by a primary instinct to seek photons. This shoot persists in its vertical climb until it breaks the surface interface. But if a seed can sustain viability for 32,000 years within glacial ice, what actually defines the outer limit of its biological clock? We are still confronted with the enigma of extreme longevity and the threshold of biological time. The first green shoot breaches the surface, and the transition from a millennia-old Siberian dormant state to a living, photosynthesizing organism is complete. The protein-rich endosperm provided the energy for the initial upward thrust, and the radicle successfully established a subterranean anchor. As the plant emerges, the cotyledons wither, and they've served their purpose as fuel reservoirs. This is the moment the biological machinery pivots. Once the internal reserves are depleted, the nascent leaves assume the responsibility of energy production via photosynthesis. These leaves capture solar radiation, transforming the organism from a consumer of stored fuel into a self-sustaining structure. A seed functions as a biological mandate, a forest waiting within a rigid, tiny husk. And finally, that mandate has breached the interface of the earth,